Inflammation. Mediators of inflammation. Mediators of inflammation. These are a large and increasing number of endogenous chemical substances which mediate process of acute inflammation. Mediators of inflammation have some common properties as under. Mediators are released either from cells or are derived from plasma proteins. All mediators are released in response to certain stimuli. These stimuli may be a variety of injurious agents, dead and damaged tissues, or even one mediator stimulating release of another. Mediators act on different targets. They may have similar action on different target cells or differ in their action on different target cells. They may act on cell which formed them or on other body cells. Range of action of different mediators are increased vascular permeability, vasodilation, chemotaxis, fever, pain and tissue damage. Mediators have short lifespan after their release. Mediators of inflammation. Cell-derived mediators. Vasoactive amines. Arachidonic acid metabolites. Lysosomal components. Platelet activating factors. Cytokines. Free radicals. Plasma-derived mediators. The Kinnin system. The clotting system. The fibrinolytic system. Complement system. Cell-derived mediators. The various cell-derived mediators are given as follow. Vasoactive amines. They give earliest inflammatory response. They are of mainly three types. Histamine, serotonin, 5-hydroxytryptamine. Another addition to this group is neuropeptides. Histamine. It is stored in granules of mast cells, basophils and platelets. Released due to heat, cold, irritation etc. Action. Vasodilation, vascular permeability increases, itching, pain etc. 5-hydroxytryptamine, 5-HT or serotonin. It is present in tissues like chromaffin cells of GIT, spleen, nervous tissue, mast cells and platelets. The actions of 5-HT are similar to histamine, but it is a less potent mediator of increased vascular permeability and vasodilation than histamine. Neuropeptides. Another class of vasoactive amines is tachykinin. Neuropeptides such as substance P, neurokinin A, vasoactive intestinal polypeptide, VIP, and somatostatin. Arachidonic acid metabolites, eicosanoids. Arachidonic acid is a fatty acid, eicosanotetranoic acid. Arachidonic acid is a constituent of the phospholipid cell membrane, besides its presence in some constituents of diet. Arachidonic acid is released from the cell membrane by phospholipases. It is then activated to form arachidonic acid metabolites or eicosanoids. Prostaglandin is major arachidonic acid metabolites. Lysosomal components. The inflammatory cells neutrophils and monocytes contain lysosomal granules which acts as inflammatory mediators. They are responsible for chemotaxis, degradation of bacteria, release of collagenase etc. On release elaborate a variety of mediators of inflammation e.g. granules of neutrophils. Neutrophils have three types of granules: primary or azorophyll granules, secondary or specific granules and tertiary granules or C particles. Granules of monocytes and tissue macrophages. These cells on degranulation also release mediators of inflammation like acid proteases, collagenase, elastase and plasminogen activator. Platelet activating factor, PAF. It is released from IgE sensitized basophils or mast cells, other leukocytes, endothelium and platelets. Apart from its action on platelet aggregation and release reaction, the actions of PAF as mediators of inflammation are increased vascular permeability, vasodilatation in low concentration and vasoconstriction otherwise, bronchoconstriction, adhesion of leukocytes to endothelium, chemotaxis. Cytokines. Cytokines are polypeptide substances produced by activated lymphocytes, lymphokines, and activated monocytes, monokines. Its major functions are adhesion and vasodilation. Free radicals, oxygen metabolites and nitric oxide. Oxygen-derived metabolites are released from activated neutrophils and macrophages, which plays major role in increasing vascular permeability, and it also includes superoxide oxygen, O2, H2O2, O and toxic NO products. These oxygen-derived free radicals have the following actions in inflammation: endothelial cell damage and thereby increased vascular permeability. Activation of protease and inactivation of antiprotease causing tissue matrix damage. Damage to other cells. Nitric oxide, NO. NO plays the following role in mediating inflammation: vasodilatation, antiplatelet activating agent, possibly microbicidal action. Plasma protein-derived mediators, plasma proteases. These includes various products derived from activation and interaction of four interlinked systems: kinnin, clotting, fibrinolytic and complement. The kinin system. This system on activation by factor Shia generates bradykinin, so named because of the slow contraction of smooth muscle induced by it. Bradykinin acts in the early stage of inflammation and its effects include smooth muscle contraction, vasodilatation, increased vascular permeability, pain. The clotting system. Factor Shia initiated the cascade of the clotting system, resulting in formation of fibrinogen, which is acted upon by thrombin to form fibrin. The actions of fibrinopeptides in inflammation are increased vascular permeability, chemotaxis for leukocyte, anticoagulant activity. The fibrinolytic system. This system is activated by plasminogen activator, the sources of which include kallikrein of the kinin system, endothelial cells and leukocytes. The actions of plasmin in inflammation are as follow. Activation of factor 12 to form prekallikrein activator that stimulates the kinin system to generate bradykinin. Splits off complement C3 to form C3A, which is a permeability factor, and degrades fibrin to form fibrin split products, which increase vascular permeability and are chemotactic to leukocytes. Complement system. The activation of complement system can occur either by classic pathway through antigen-antibody complexes or by alternate pathway via non-immunologic agents such as bacterial toxins, cobra venoms and IgA. Complement system on activation by either these two pathways yields activated products which include anaphylatoxins, C3A, C4A and C5A, and membrane attack complex, MAC, i.e. circa 5B, C6, 7, 8, 9. After activation it shows following actions: release histamine, increased vascular permeability and increased phagocytosis.